With our knowledge of nucleic acids, let's get to apply that in HIV and cholera disease. Now, starting with the HIV, which is the human immunodeficiency virus, this one is a retrovirus, and the meaning of that is that the genetic material that HIV has is in form of RNA, and it is going to use this RNA to go to DNA. This DNA that it is going to form is now going to join with the DNA of the host. So if this virus is in a person, when it converts its RNA into DNA, that DNA that it forms, let's say it forms this DNA, is going to be embedded or is going to merge with the DNA of the host, that person. And when this happens, this is DNA again, this is going to now be converted into RNA, and then we are going to be able to form proteins. So at the end, you are going to have proteins which have got also the genome of the virus itself. And so this will continue replicating, and it might be very difficult for you to be able to remove it. That is why this is also very difficult to get to treat. Apart from that, HIV virus is very hard to treat in that it undergoes mutations. When you want to deal with one form of the virus, by the time you wanted to make a medicine to deal with that, you are going to see it changes into another form that you are not able to recognize. So we are going to start with this. This is the basic structure of the virus. We have got the outermost part. We call this the envelope of the virus. Now this envelope at intervals, it has got glycoproteins. So for me, I'm going to use the same glycoprotein. We are just going to use GP to represent that in a short form. So that is glycoprotein. Now it has got two forms. On we have got this glycoprotein, which is a transmembrane protein, meaning it penetrates the cell wall of, let me say, the cell membrane of this virus. So it goes through. And then on top, we have got another one which attaches to that other one which penetrates. So this one, which is on top, binds together with the one which is a transmembrane. So they'll continue like that for all these others. So the one which is on top, what happens is that for in our cells, in our bodies, we have got macrophages and also CD4 cells. Now these are used as fighters in the body. So for the CD4, we have got what we call CD4 receptors. So if this is the cell membrane of a person, there are CD4 receptors. These are the ones which make sure that if there is a pathogen or anything that doesn't belong to the body, which is passing in the body, the CD4 receptors are able to track that and then be able to communicate with other molecules to come and destroy such a pathogen. Now, what happens is that the same CD4 receptors, of course, we are saying these are receptors, meaning that this is where the CD4 molecule is able to attach. And the CD4 molecule is there to make sure that it invites other molecules to fight uh, pathogens. Now, this same CD4 receptor can also attach this virus. So, this part of the virus is able to attach to the CD4 receptor. And in that way, we are going to see that at the end, as it goes on, when this portion of the virus attaches to the CD4 receptor and remains there, in time, this cell membrane is going to become, let's say, a bit of less permeable, and it is going to allow this molecule to be able to pass or enter inside. Now, that doesn't just happen because there are some mechanisms that the virus is going to use to make sure that it penetrates the cell. Now, we are still understanding the structure of this virus. So, this portion which is able to attach to the CD4 receptors of the host, this one, remember we said this is the glycoprotein. So, the top part we call this GP120, the glycoprotein 120. The 120 is simply representing the weight of that glycoprotein. And then the one which is down, which is the transmembrane protein, this one is called the GP41. So I've got the GP120 and the GP41. All right. Now again, just below this, we call this as the envelope. Just below the envelope, we have got something else. So we're going to have something just below the envelope like that. And we call this as a matrix protein. So this matrix protein, because it is a protein, we are just going to give this a P. Now this one, we call it a P17. So that matrix protein, which is just below the envelope, is called the P17. And then inside this matrix protein, we have got also some other structures. So we have another structure, which is going to look something like this. So this structure is the one which contains some other structures which are found in DNA. This is actually the one which contains the RNA or the genetic information of the virus. Now, what is the name of this structure? This is called the capsid. So that is the capsid. Now, inside the capsid, we are going to have two strands of RNA. So these are the strands of viral RNA. Another thing that we have inside here is that we have got three proteins. So let me say we have got one there, we've got one here, three different proteins. So the first one is called reverse transcriptase. And the next one from these three proteins is called integrase. And then the other one is called protease. Now, why do we need to have all these proteins? Well, we're going to understand why we need to have all these proteins in this virus. This is just the structure of this virus. So basically, this is the basic structure of this virus. Inside the capsid, we have got two RNA strands. Now, just these RNA strands, they are found in what is called the nucleocapsid. What is the nucleocapsid? The nucleocapsid just contains the RNA surrounded by proteins. So what we are going to see is something like this. This is the RNA strand, and then it has got some proteins. So that is a capsid. Okay. So this is a basic structure. So what happens is that the GP120 is able to attach either to the macrophages, which are the fighters in our bodies, or they can attach to the T cells, which are the thymus cells. Now, depending with where they are attaching, they are able to attach to so just on the cell membrane, we said we have got CD4 cells. CD4 cell receptors. Now, this virus is supposed to attach to the CD4 cell receptors. It's not only this structure, there is also another part which we call the core receptor, which works together with the CD4 receptor. Now, the core receptor in the macrophages, we call it the CCR5. Okay, I've put on the T cells. So the core receptor in the macrophages is called CCR5, and then the core receptor in the T cells is called CXCR4. So this portion, the GP120, is going to be able to attach to the co-receptor and the CD4 receptor. So that is basically what gets to happen in this process. All right. So now we have understood this. Let's continue and go on with the structure of this virus. Okay. So this is basically what I was just trying to draw. We have got the envelope outside, and then we have got the HIV glycoprotein, which we are able to see outside here. The HIV glycoprotein has got this portion which is outside, which is the GP120, and this one which is the transmembrane which goes inside the cell membrane, which is a GP41. And then we have got inside the HIV capsid, which is this molecule which you're able to see here, or this portion which you're able to see. And then inside the HIV capsid, we have got HIV enzymes and also the HIV RNA. And we have got forms of the two strands of the HIV RNA. So the HIV capsid is the HIV's core, that is a center that contains HIV RNA, as we're able to see, and also some enzymes, specifically those three enzymes that we mentioned. So the HIV envelope, which is the outside, is the outermost layer of the HIV. And then we've got HIV enzymes which are found inside. These processes are going to make sure that they carry out steps in the HIV life cycle. And then we have got HIV glycoproteins, which are basically protein spikes. These are the ones which are able to see outside here, which are embedded in the HIV's envelope. And then we have got the HIV RNA, which is the genetic material of the HIV. So again, this is the structure. Now it is just a bit more in detail. We can see we have got the GP120, which attaches to the CD4 receptors. And then we have got the GP41. Now, what is the function of this GP41? We know the function of GP120. What of the GP41? So this GP41, we can see it gets to go inside of the cell, like it is a transmembrane protein, right? So the GP41, of course, is there to make sure that it supports the GP120. We can see if there was no GP41, where would be the GP120? It wouldn't attach anywhere. So the GP41 is basically there for the attachment of this GP120. Now inside we have got this structure just after the envelope, and this structure just outside the envelope is what we call the matrix protein, and it is made up of P17 or protein 17. And then we have got also this other structure. This structure is what we are calling the capsid. Now this whole structure which we are able to see, this thing which is outside the envelope, is a lipid bilayer. It actually comes from the host. The HIV virus itself doesn't have this lipid bilayer, so this one came from the host. It just took it from the host as it was attacking the host, right? So the capsid contains these structures which are inside. We can see we have got the nucleocapsid. The nucleocapsid is the one which contains the RNA and also some proteins. So we can see the reverse transcriptase there. We can see the integrase, and there are also some other proteins. Okay. So we also have the protease there. Now, all these structures that we are able to see, they are inside here. So this is basically the structure of the HIV virus. All right. So again, this is the same one. We can see the envelope surface that has got GP120, that is the outside, and then we have got the transmembrane glycoprotein, which is GP41, and then we have got GAG, membrane associated matrix protein, which is P17, So just inside here, we have got what we call the membrane associated matrix protein. We already said we have got a matrix, which is a protein which just comes below, just below the envelope. And we already mentioned that was P17. And then we also have capsid, which is on the core, this center. The capsid is made up of P24. P24 protein 24 is what makes up the capsid. And then inside the capsid, you have got two molecules of RNA, and then we have got proteins. So basically, that is the structure. Now, what is GAG? What does that even represent here? Well, this GAG is just a group of specific proteins, which are the major structural proteins. So if we just hear of GAG, that's the major structural protein of the HIV virus. So you can see GAG, which is the matrix protein. We can also again see GAG, which is the capsid. So that is just a structural protein of this virus. Now let's look at the life cycle of the HIV. So as we said, we can see the HIV will face is that it is going to bind to the receptors on the cell membrane of the host. So immediately the HIV virus enters the body, it is going to attach to the receptors inside the host, the cells of the host. So we are able to say this basically, this receptor here should be CD4 receptor and the core receptor, depending with whether this is a T cell or the macrophages, because those are the ones which the HIV virus is able to attack. If it is a T cells, we're going to have the CXCR4 core receptors. If it is the macrophages, we're going to have the CCR5 core receptors. So the first thing that the HIV virus does is that it's going to bind to the receptors. When it binds to those receptors, it's now going to fuse with the cell membrane of the receptors. I mean, with the cell membrane of these host cells. When it does fuse, what is going to happen is that the whole of this structure of the virus is going to remain embedded in with the cell membrane of the host, and then only the proteins which were inside the capsid and also the RNA is going to go inside. And then step 3, remember we have got 3 enzymes inside of the virus. We have got the reverse transcriptase, we have got the integrase, and we also have the protease. So what the reverse transcriptase does is that we know that according to the central dogma of molecular biology, DNA is supposed to convert to RNA and then RNA to proteins. Now because HIV has got RNA, we want to convert the RNA of the HIV to DNA. That is why it is called reverse transcription, because transcription itself is supposed to convert DNA to RNA. So for the reverse transcription, the reverse transcriptase enzyme that is part of the HIV is going to do that work. It is going to convert the RNA into DNA. And then the next thing that happens is that when you have got now the DNA, we call it the viral DNA. That viral DNA is able to now integrate or join with the DNA of the host, and that is called integration. And the enzyme which does that is the integrase. So viral integrase is going to make sure that the DNA of the virus and the DNA of the host integrates to become one. And when that happens, replication is going to happen. So this is going to make a copy. So the DNA of the host and the DNA of the virus is now going to make a copy like that. And then there's going to be assembly. Now, after making this copy, these are going to be assembled, the DNA of the host and the DNA uh, of the virus. And that will continue, and proteins are going to be made. Remember also of the protein, the protease that is part of the HIV. So the protease is going to make sure that proteins are formed. And when that happens, then this is going to be assembled. Again, just the cell membrane of the host, when the viral DNA reaches that point, is going to bud off in what is exocytosis. So it's going to be removed outside the cell, because at the first point there was endocytosis. and then this is exocytosis. So this is going to be removed outside the cell. That's why we say the envelope of the HIV is not its own envelope, but it comes from the host. That is why this envelope is also a lipid bilayer that is going to come outside the cell. And this will continue replicating and replicating until they become so much many inside the cells. So this is what gets to happen. Now, we have got structures which are called nucleoside analogues. What are nucleoside analogues? Now, we know when we are talking about a nucleoside itself is simply a structure that contains a pentose sugar and a nitrogenous base. A pentose sugar and a nitrogenous base. Well, these nucleoside analogues, they are simply structures which resemble nucleosides, meaning that it also looks like this nucleoside which is found in the body. We know that in the body we have got a lot of nitrogenous bases and also the pentose sugar. If we are making DNA, the pentose sugar that we're supposed to have is deoxyribose. If we are making RNA, the pentose sugar that we're supposed to have is ribose. And then in humans, or let me say in animals like ourselves, when we are forming this DNA is going to have four nitrogenous bases. We have got adenine, cytosine, guanine, and thymine. We don't have uracil in DNA. We won't have uracil in RNA. So these nucleoside analogues, they resemble nucleosides. Now, because this is a nucleoside analog, we are able to use these in forming medicines. Because they resemble the nucleotides, what happens is that this HIV is supposed to be attacking this DNA of the host. Now, if we have got these nucleoside analogs, now because this is a nucleoside, DNA is made from a nucleotide, meaning a nucleoside plus a sugar, I mean a phosphate group. That is now going to make what we are calling as the nucleotide. Let me see if I'm able to write this. Okay, maybe I'm just able to show this. What we have here is that we have got this compound here, which is a sugar. Now, in this case, this is a ribose sugar because we can see we have got OH at position number 1 and position number 2. And then we have got the base this side, and then we have got 3 phosphates. Now, if I remove these phosphates which are here, then what I'm having is a nucleoside. If I add the phosphates, then I have a nucleotide. So the first thing that is going to happen is when we're talking about nucleoside analogs, we want to change from nucleoside to nucleotide. And we are going to perform what we call phosphorylation. So to a nucleoside, if we add a phosphate, we are going to form a nucleotide. That is what we call phosphorylation. And so what is going to happen is that at each stage, these nucleoside analogues, which we have phosphorylated to form a nucleotide analog, a substance which resembles a nucleotide, at each point they want to fight the virus. So just at the first point here, when the virus is binding to the receptors, they are what we call binding or fusion inhibitors. So we have got these medicines which are able to take, which are going to bind to these receptors such that it prevents the HIV from binding to these receptors. So in such a way, we are able to prevent the accumulation of these HIV viruses. Now, in a case when the HIV has already bound and then it goes inside the cell, the next thing that happens is that the RNA of the virus is supposed to go into DNA. So in such a case, we have got the reverse transcription that is happening. So we have what we call reverse transcriptase inhibitors. There are also some other analogues which are able to fight the conversion of the RNA to DNA, because we know that what converts RNA to DNA is the reverse transcriptase. So these are going to be able to fight the reverse transcriptase. So we call these inhibitors. Now, in a case where still the RNA has been converted into DNA, the next thing that happens is that the DNA that has been formed of the virus is supposed to integrate with the DNA of the host. So there are also, we know the enzymes which do that is the integrase enzymes of the HIV. So they are also integrase inhibitors. So those are there to make sure that they avoid or they prevent the joining of the viral DNA with the host DNA. So in case it still happens like that, there are still others, because if the DNA of the virus is able to integrate with the DNA of the host, transcription is then going to happen. Then you are going to form again RNA. And then when you form RNA, you're again going to form DNA in, I mean proteins in what we call translation. And then after then, these proteins are able to move out. Now, before you're able to form these proteins, there's also another enzyme called protease inhibitor. So these inhibitors, they want to prevent the formation of these proteins, or they want to prevent the function of these proteases. So these are nucleoside analogues. These are medicines which are able to be given at any level to just make sure that HIV is fought. So this is also just explaining what we're from talking about. So the stages in the life cycle of HIV is that you have got the HIV first attaching to the CD4 receptors, as we're able to see at this stage one. And at this point, we are able to fight this. So of course, we have got also co-receptors. We already mentioned that the CXC, we have got the CXCR4 and the CCR5. And then the next thing is that HIV enters a CD4 cell. Okay, it is going to enter the CD4 cell, and then the capsid enters the nucleus where HIV proteins and enzymes are released, the nucleus of the host. And then the process continues like that as we've described. Now let's look at cholera. How does it happen then in cholera? Now, cholera is caused by what is called Vibrio cholerae. That is the causative for cholera. Now we have got what we call cyclic adenosine monophosphate. What is the function of the cyclic adenosine monophosphate? So what the cyclic adenosine monophosphate does is that this one, okay, let me first look at this structure. Cyclic adenosine monophosphate. Now we are saying adenosine monophosphate. We have got first the pentose sugar here, and then we are going to have a nitrogenous base here at position number one. Now this is adenosine, meaning that this pentose, this should be adenine, the nitrogenous base. Now we have got monophosphate, meaning we only have one phosphate. Now because this is cyclic, what happens is that this phosphate is going to attach like this at position number 5 and position number 3. And so it forms a circle like that. So it connects from position number 5 and position number 3. This forms what we call cyclic adenosine monophosphate. Now, the cyclic adenosine monophosphate has got functions in the body. Cyclic adenosine monophosphate regulates cell membrane permeability. So it is responsible for the permeability of the cell membrane. Apart from that, the cyclic adenosine monophosphate also modulates transcription and translation. So it has got a lot of functions. So in the permeability, it is involved in the permeability of sodium ions, calcium ions, potassium ions, and also water in the body. So then what happens? So this enzyme we have, I mean, this causative of cholera, which we have called Vibrio cholerae, is going to release a toxin. And that toxin which Vibrio cholerae releases activates the enzyme adenyl cyclase. So this enzyme here, adenyl cyclase, is activated. Adenyl cyclase converts ATP, which is adenosine triphosphate, to cyclic adenosine monophosphate and also two phosphate groups. Because this was adenosine triphosphate, we add three phosphates. So two of these phosphates are going to be cut off, and then one is going to remain. And then this one which remains is going to connect to the third position of the pentose sugar to form cyclic adenosine monophosphate, and then we have got also two phosphates. Now when we form this cyclic adenosine monophosphate, we are increasing the adenosine monophosphate inside the cells. So then what is going to happen? Because we have increased the adenosine monophosphate, this is going to alter the sodium and chloride transport, because just inside in the cell membrane we have got these pumps, the sodium pumps. We have got a lot of pumps. So, so what happens? Let me try to show maybe. Okay. So if we have got, let's say, the cell membrane, we have got all these pumps which allow the intake of the sodium and the intake of chloride ions. Now, the way we know is that the sodium is more. If we take it, potassium is more inside the cell, sodium is more outside the cell. Chloride ions are also more outside the cell. Now, because we have disturbed or we have increased cyclic adenosine monophosphate, what this does is that it is also going to affect the chloride channels. Now, because it has affected the chloride channels, what is going to happen is that the chloride channels are going to open. And so, if we have got the stomach, okay, we have got the stomach. How am I even going to be able to show that? Okay. Okay, let's say we have got the lumen of the stomach, that open portion of the stomach. So because we have opened these chloride channels, because cyclic adenosine monophosphate is there for permeability of the cell membrane. So when the chloride ions are moving from the outside of the cell to that space, which is the space of the stomach. When that happens, every time chlorine is moving, it attracts water with itself. So there is going to be an increase of water in the lumen of the intestines. And because of that, that is going to be able to cause diarrhea. So cholera actually causes diarrhea because it affects cyclic adenosine monophosphate, which opens the chloride pumps, and chlorine moves to the lumen of the intestines, attracting water with itself. And so, because of the increased water volume inside the lumen, that causes diarrhea. So, that is it for this tutorial. Thank you so much for watching.